Hello students, now we are going to discuss about Drosophila transposons. It is estimated that 15% of the genome of Drosophila is mobile, so it contains transposable elements. So there are two classes of transposons which are observed in Drosophila. The first one is copia element and second one is P element. So now we will discuss P element in detail. So P element is containing IR at both the ends and it is also flanked with LTRs. IR is 17 base pairs and LTR, that is long terminal repeats. So copia element is having an additional sequence of LTR. So copia element is flanked by both the ends. It is flanked by LTR, long terminal repeats, and then it has IR sequence. The total number of base pairs in copia element is 5000 base pairs and LTR is 276 base pairs and IR is 17 base pairs. So coming to the transposition of copia element, how it is moving from one place to other. Copia element shows retro transposition, that is it is moving via RNA intermediates. Copia element first it synthesizes mRNA, then mRNA will be reverse transcribed to DNA and then that DNA will be inserted into a new location. So that's why it is called as retrotransposon. Copia element is a retrotransposon. Why? Because it is moving via RNA intermediate. And then this copia element, whenever it is moving, it during transposition, copia element is generating target site duplication, that is it is generating direct repeats. The number of base pairs which are formed, that is target site duplication, is 3 to 6 base pairs. Then coming to the number of copies of copia element in the genome is 5 to 100. 5 to 100 copies are present per genome. The second class of transposon in Drosophila is P element. P elements, there are two types of P element. P element, complete P element, and second one is incomplete P element. See the size of the P element here. Complete P element has a IR at both the ends, inverted repeats. The number of base pairs in the P element is 31 base pairs and the total number of base pairs in the P element is 2907 base pairs, that is 2.9 kb DNA. Okay, if there is any deletions in IR, that is if there is any deletions in the P element, it produces incomplete P element. So here P element, complete P element and incomplete P element. Complete P element is able to synthesize transposase enzyme, so it can move from one place to other. So that's why this complete P element is an autonomous element of the family. It is able to move from one place to other. Okay, the second one is incomplete P element. Incomplete P element has been produced by deletions. Internal deletions in the complete P element results in the formation of incomplete P element. I repeat, incomplete P element is formed due to internal deletions in complete P element. Okay, if there is any deletions in this complete P element, will result in the formation, that is it produces or it generates incomplete P element. Incomplete P element, it cannot synthesize transposase enzyme. So functional transposase enzyme cannot be produced by incomplete P elements. So that's why incomplete P elements are non-autonomous element. On its own, it cannot move from one place to other. Okay, due to internal deletions, it cannot synthesize transposase enzyme, so it is not moving from one place to other. But complete P element provides transposase enzyme which is required for the transposition of incomplete P element. So complete P element will provide enzyme transposase enzyme which is required for the transposition of incomplete P element. So in presence of complete P element, incomplete P element can move from one place to other. So now coming to, this is the structure of complete P element and incomplete P element. Coming to the existence of this P element, like P elements are not found in the flies which have been captured before 1950. So 1950, before 1950, they do not have P elements, that is Drosophila do not have P elements, and these Drosophila are called as empty strains. Empty strains are nothing but Drosophila without P element. So but these P elements have invaded Drosophila. Okay, so P elements before 1950, the flies which have been captured before 1950 do not have P elements and they are called as empty strains. But later the P elements have invaded the population of Drosophila. So this P elements, how they entered into Drosophila is not clearly explained, but the speculations are that P element might have entered through virus. So now causes of P element. The P element causes hybrid dysgenesis. Hybrid dysgenesis is nothing but a series of defects that results from crossing certain Drosophila strains. P elements causes hybrid dysgenesis, that is nothing but a series of defects that results from crossing certain Drosophila strains. Coming to the defects, the defects can be like mutations, chromosomal aberrations and sterility. The sterility, all these like defects will be considered as hybrid dysgenesis. So here we will discuss two different types of cross. Okay, this is the first cross, M cytotype and P cytotype. M cytotype is without P element, P cytotype is with P element. So before going to this cross, you need to understand flies are the cell which has P element. If P element is present, P element encodes for a repressor protein. P element encodes for repressor protein which inhibits the expression of transposase gene. So transposase enzyme will not be synthesized in presence of repressor protein. So in P cytotype, P elements are stable. In P cytotype, even though they have P element movement is blocked in P cytotype. But whereas M cytotype, they don't have P element. And the second one is, if the P element is not there, there is no repressor. Okay, so remember, the first cross we will discuss M cytotype and P cytotype. M cytotype is without repressor protein and P cytotype has P elements and it has repressor protein also. Okay, so now when M cytotype female is crossed with P cytotype male, so what happens in the F1 hybrids we will discuss. F1 hybrids, they look normal but the flies are sterile. Why the flies are sterile means M cytotype is not having P elements. Okay, P cytotype has P elements. So cytoplasm in a cytoplasm M cytotype is not having repressor protein. When P elements are entering into M cytotype, repressor protein is not there. So when repressor protein is not there, P elements will synthesize transposase enzyme. So because transposase enzyme is been like synthesized, P element will move. Okay, so P element whenever it is moving, okay, it causes hybrid dysgenesis. This movement is observed, or hybrid dysgenesis affects only in the germline cells. Okay, that's why Here we can see the flies are sterile because of hybrid dysgenesis and there is no progeny. So remember, M cytotype is not having P elements and repressor protein. P cytotype is having P element and as well as repressor protein. Repressor protein blocks the expression of transposase enzyme in P cytotype and prevents the movement of P element in the P cytotype. But in this cross, P elements are exposed to M cytotype. M cytotype is not having repressor protein. Because of lack of repressor protein, transposase enzyme is synthesized, okay, which causes the transposition of P elements and that leads to hybrid dysgenesis. So this is very simple concept, okay, try to remember. Okay, next is the second cross. Second cross is P cytotype female and M cytotype male. So in this cross, P element is present in this P cytotype and repressor protein is also present. In M cytotype, P element is absent and repressor protein is absent. Now in this cross, FN hybrids, they are normal appearing progeny and the cytotype which we observe in the FN hybrid is P cytotype. So now P elements are present in P cytotype. So P cytotype repressor protein will be present. So repressor protein, that is P cytotype repressor is present. So it prevents transcription that is expression of transposase gene. So transposase gene is not expressed, the enzyme is not synthesized, so which leads to the block in the transposition of P elements. I repeat, P elements in P cytotype are stable. Okay, why in P cytotype P elements are stable is because of presence of repressor protein. In this class, P elements are present in P cytotype. So in P cytotype, because of presence of repressor protein, okay, this repressor protein will binds to P element and it interacts with inverted repeats and it blocks the expression of transposase gene. Okay, so repressor protein prevents the expression of transposase gene. So transposase enzyme is not synthesized. Lack of transposase enzyme prevents the transposition of P elements. So P elements are stable, so there is no hybrid dysgenesis. Okay, flies are fertile. So the difference between the P cytotype female and M cytotype male, and then here M cytotype female and P cytotype male is here the P elements are present in M cytotype, so lack of repressor protein, expression of transposase gene. Then you will observe transposition of P element. Here P elements are exposed to P cytotype and P element is stable. Okay, hybrid dysgenesis is observed only in the first cross, that is M cytotype female when it is crossed with P cytotype male. And remember, M cytotype male or female flies are not present naturally. Okay, they are called as mutant lab strains. So these are mutant lab strains. M cytotype female or male will be considered as mutant lab strains. So remember, there are two different types of transposable elements in Drosophila, copia element and P element. Copia element is only single type. Okay, it is a retrotransposon. And here P element has two different types, complete P element and incomplete P element. Complete P element is autonomous element. Incomplete P element is deletion derivative of complete P element. So incomplete P element is non-autonomous. And then coming to the P element, how it has invaded. Okay, so it has invaded via virus. And then we need to talk about what is hybrid dysgenesis.